So I just wanted to welcome everybody to the Lasker Lessons in Leadership. This is, I think, our 9th lecture. This was started in 2015 it's a collaboration between the Albert and Mary Lasker Foundation, the International Biomedical Research Alliance, and the NIH Oxford Cambridge Scholars Program. My name is Randy Belletta, and I think most of you know that. And I just wanted to welcome everybody tonight, and I hope that you'll find this enjoyable and that you'll learn something. I also wanted to very quickly thank our sponsors who have been, I think, with us from the very beginning since we started this. Auerbach Schrott, and we have Jeff Auerbach here. Where are you, Jeff? Okay, thank you very much. And Certara and WCG have been supporters of this event since this was begun. So I'd like to introduce Steve McLean, who's the chairman of the Alliance Board, and he's going to say a few words and then introduce Dr. Yancopoulos. Thank you.
Thank you very much, Randy. It's a joy to be here tonight. Great to see that actually Washington can handle a blizzard, so congratulations for you all to be out here tonight. It's also a personal privilege tonight to be able to introduce one of my idols. And you know, it's such a rare opportunity that you get to do that these days that I want to thank Randy, the Alliance, and the Lasker Foundation for their insight and genius in picking George to be the speaker this evening. You know, George in so many ways personifies the kind of person, the kind of professional, the kind of scientist that is the fondest hopes we have for the program and for each one of you individually. You know, George is an MD, PhD with a deep understanding of human biology. He is also a gifted scientist who had the foresight to take that understanding of human biology, apply it in a very novel way to human disease conditions, and especially early on, a specific human disease condition involving adult macular degeneration, and to be able to develop a new model of using the human angiogenesis to be able to treat that using a VEGF medication, therapeutic. And it was a wonderful scientific achievement. But he also is an innovative and inspirational entrepreneur, because in his pursuit of science, he helped to build a wonderful company, Regeneron, which is known not only for the therapeutics it has developed over the years, but also for the platform that it has developed. The fact is that Regeneron, following in George's model, not only focused on therapeutics, but on the tools and the processes to continue to develop additional therapeutics to ease human suffering. And they continue to be leaders in all facets. In fact, the most recent collaboration that now goes back 4 or 5 years with Geisinger, where they are very innovatively intersecting both the phenotype and the genotype of the patients. That unique population in Western Pennsylvania shows is a portent of future discoveries of therapeutics into the future. And so, without any further ado, it's my pleasure, my honor to introduce my friend George Yancopoulos, who, by the way, I want to just add one other thing. Not only from an organizational point of view have they focused on therapeutics, but it also has become, if you will, like the Disneyland of science, that one of the happiest places on earth to do science, because they've followed the traditional virtues of doing well while doing good. And you see that in the commitment that George and the senior executives make every day to all sorts of philanthropies, not the least of which is their involvement with us here at NIH and at the Oxford Cambridge Scholars Program. Thank you, George.
Well, thank you, Steve. But now I got nothing left to say. He said it all. I also got to say I'm going to call you out because we were just talking about it. I am not his idol. Both of our idols is Roy Vagelos. Yes. And if you guys don't know who Roy Vagelos is, buy the book and read it, because that's all I did, and that's why I am where I am today. So I just recommend finding out everything you can about Roy, read his book, and just follow the recipe that he literally wrote in the book on how to do science to come up with cures. And I'll give you our sort of attempts to do, you know, what Roy taught us. Roy, by the way, is our chairman. So of course, we follow, we're trying to follow in his footsteps. I also have to say, I am not here for you. I'm shameless about it, open about it. I'm here for me, okay? My whole goal is to just try to get some of, we don't have to get everyone, some of the best and the brightest to come join us. As you heard from Steve, you may or may not know this, but for 6 out of the last 7 years, we've just been selected the best place in the biopharma industry to work. Okay, so we are a great place, I think. Which is pretty amazing, because there's 5,000 biotech and biopharma companies in the United States alone. So being number one, 6 out of 7 is pretty. The only company that was sort of like us before us, which was sort of our model actually, was Genentech in its heyday. And obviously things have changed there, which is, I think, a little, what's going on, Randy? Oh, I got this, so this is redundancy? I don't know. Now, what do I do with my right hand? Okay. So, yeah. So, and I do think that, you know, the world needs, I mean, the great Genentech at its peak was a great place, and, you know, we need more places like that. Unfortunately, there aren't as many as we would like. So I am going to tell you, while shamelessly trying to recruit you, you know, about why we're such a great place to do science, because we really truly do exactly what Steve was saying. We try to take advantage of technologies, mostly genetics-based technologies, to understand and define the best targets for drug development, and then we develop other technologies to give us the drugs, and lo and behold, it actually works. And very few people have actually taken this approach, as I'm about to tell you. So the first thing that I thought I had to do was to tell you a little bit about our industry. I know some of you were at Regeneron. How many of you guys were at Regeneron recently? Okay, good. So this won't be repetitive for all you guys. Maybe some of you guys heard it. Usually, I apologize for not being able to meet with you guys. I like to meet with you guys because I just want to recruit you guys. You know, when you guys come up to Regeneron, but something must have been important. I can't remember what it was, though. Came up that day. So the first thing that I'm going to do is tell you about our industry, which I did not know when we started Regeneron. And in fact, frankly, if I knew this, I would probably not have gone into biotech, because I would not have understood how challenging it is. I do think things are changing, and maybe this is a great time to go in. Okay, so by the way, this is our 30-year anniversary. Our goal since the beginning was used to the power of science, and particularly genetics, to bring new medicines to patients, and not just once, over and over again. It's still astonishing to me that almost nobody does this to this day. Genetics, I mean, genetics is the basis of life. DNA determines like so much, and most people don't focus on it. I'll tell you what they focus on. They focus too late in the process. So I think we would all agree. I mean, I know I got hooked on science when I was really young, and by the time I was in high school, I wanted to be, you know, a life sciences researcher. I was really into biology and chemistry and all that. And I imagined, I read all the books about all the people who had done great things. I just wanted to use science to come up with something really important to treat human disease. That was, you know, my dream. And I think a lot of you guys probably were just like that. I think a lot of us who go into science do that. And the thing they don't tell us, certainly not in high school, not even college or graduate school, is while I think we all agree that inventing new medicines, important new medicines, is among the most important things that we do as a society, because we have so many healthcare challenges, most people don't realize the odds against us, and the enormous time, costs, and efforts, and the sobering results. And like I said, if I actually knew this when I started out, I would like, wow, gone into something else. But basically, just in the United States, there's about 5,000 biotech companies, not to mention all of the outstanding institutions, academic institutions, from the NIH to Harvard to all sorts of other places. It involves millions of researchers worldwide. Hundreds of billions of dollars are spent just on the R&D side by the biopharma industry. And despite all this, almost everything that we do, starting in most cases from academic research, the NIH-funded stuff and all that, through biopharma R&D, almost everything fails. And this is almost an underestimate because this is of things that have entered into clinical trials already. So this does not include everything that fails to get into clinical trials because the data is not there. And all of this huge effort, there's only 20 to 40 new drugs approved by the FDA every year. I mean, that's really, for all this effort and everything that we all do, 24 new drugs. It's even worse than that, okay? Most of these are Me Too's, okay? Something that's just sort of like a follow-on or, somebody trying to get in on another big opportunity. Why do they do that? Because that's a lot easier than coming up with a first-in-class. So every year for the last 10, 20 years, there's only 5 to 10 first-in-class medicines. It's even worse than that. Nothing wrong with this, but most of these are for rare diseases. Why? Because the whole process is a lot easier for rare diseases. The number of studies you have to do, the number of patients you have to treat, the regulatory issues, how much safety you have to get, it's just a lot easier. So every year, if we have one great new drug for a major disease that's first in class, that's good. It's usually no more than 1 or 2 a year and sometimes less, as a complete societal worldwide effort. Think how sobering that is. Okay, and unfortunately, most companies, especially the biggest companies, have given up. So now the pipeline comes from, the collection of the thousands of small companies. Most of them have an idea. 99.9% of those things fail. But 1 in 1,000 makes it to the point that a big company then says, oh wow, this has a chance at a slightly later stage, we'll buy this. That's how most things happen. The big companies have essentially almost given up on research, and their whole business plan is, let's buy the best little thing that's out there, and then by the power of our commercial organization and so forth, we'll help turn it into a big success. Okay, that's not what we wanted to do. We wanted to change the paradigm. And before I get to, sorry, a little out of sequence, what we want to do, I just want to add something else about all this, which I feel very strongly about. I mean, all over the news, you hear about drug prices, and we all want universal health care, and we all want free medicines, right? Do we really want free medicines? Do we really want low drug prices? Okay, if you sort of think about it, okay, the world doesn't appreciate, society doesn't appreciate how far away we are very, very far from having effective treatments and let alone eradicating the most prevalent diseases facing mankind. In the United States, let alone worldwide. Number 1 killer is still heart disease. Nowhere near curing that. Cancer, obesity and type 2 diabetes, a big growing epidemic. Nothing really there. I mean, other than insulin and insulin-like stuff, no real advances, certainly not for obesity. Alzheimer's, nothing. Nobody believes there's anything in anybody's pipeline that is going to even have a shot at working in Alzheimer's. In order for it to work 10 or 20 years from now, it would have to be in the pipeline today. Blow your mind. The estimates are in 20 years there's going to be 4 million Americans. Now wait, sorry, I got that wrong. 40 million. 40 million Americans. The numbers are crazy. It's a big, big number. Society can't handle these numbers. It can't handle these numbers of people with Alzheimer's, let alone obesity and type 2 diabetes, which is going to be half the U.S. population. And we got nothing in the pipelines as an industry, or very little. Okay? And we have to understand that what we should be doing is creating the biggest incentives. Okay? We should be figuring out how to, I was talking with somebody, can't remember, sorry, the name, but I was talking with somebody before, and they were talking about how, there's issues, and, why do people, go into different careers, and, the reward system and all that. We should be incentivizing the brightest minds in America to be going into this business, because what the world doesn't understand is that, forget universal health care, forget cheap drug prices, no economy, no society will be able to sustain the amount of disease that is coming upon us. And there should be nothing other than maybe climate change, okay, and world peace. But those three things, I mean, world survival, human society survival depends on this. And we should be thinking more about how to incentivize and invest in healthcare solutions and optimize finding new treatments. So I just think it's a really important thing for people to realize. We could make all drugs free today, and what does that mean? That means we're going to die of all these diseases tomorrow, and it's really as simple as that. And there's no other way, I don't think, right now to figure out how to address the problem. Okay, so we wanted to contribute to this process. We wanted to build a different type of company, and we started by, it's the only major biopharma company in the world that was started and run by scientists. Okay, normally commercial and salespeople are in charge of these companies. We think it's as Roy Vagelos. So the most successful company in the history of the industry was Merck, run by Roy. And he said, well, if you're trying to come up with cures and you're using science to do it, then you should have somebody who knows about that in charge. But most other people didn't listen to him. We listened. Okay, and our goal was to have the ability to go from an idea to important new medicines, not once, but over and over again. And like I said, we bet it all on genetics, which was obvious to us. That's where life starts. I mean, that's where we should start. Most people focus, like I just said, the big companies, they look for that product. Okay, they don't look about building a system that will lead to many products, which was our strategy. And in terms of genetics, as I'll show you, it was first large-scale mouse genetics, and now, as Steve already told you, it's large-scale human genetics. So it took us a long time. None of this was easy. But as I'm about to tell you, the last few years, people have recognized that maybe this is the way to actually build it. So when I say we, it started out with this guy here, my buddy, Len Schleifer, and myself, about 30 years ago now. And the amazing thing was we managed to convince and attract a whole series of world-class scientists to join us. And the best evidence that we're really the best place to work is that our best people, they never leave. Why? Because why go anywhere else? Because anywhere else you go, you can't do as good and as powerful sciences. So almost every one of the major strong scientists that we've ever brought to us and become part of our leadership team is either still with us or, unfortunately, a couple of them retired. We've gone from a couple of guys to over 7,500 people, more than half of them in R&D, and we've produced, what we talked about. In that, unlike we've bucked the trends, our success rates are 10 to 100-fold those of the rest of the industry. Over the last 5 years, we've gotten 7 FDA-approved medicines, many more, over 24 in clinical trials, and they're all from our own laboratories, all from our own work. We don't in-license things or whatever. These are all homegrown products. The leading drug that Steve mentioned for the causes of blindness in the United States, macular degeneration, diabetic eye diseases, and so forth. The first drug after the statin class that was discovered by Roy Vagelos, the first new way of lowering cholesterol to actually prevent heart disease and actually prevent death to a similar level like statins, but on top of statins. Important drugs for allergic diseases, which I'm going to tell you a little bit about that one, and so forth and so on. And it all started with like I said, the mouse and the human genetics. And the thing that I'm proudest of, other than the fact that, Science Magazine has now ranked us the #1 place to do science in the biopharma industry, perhaps I'm actually prouder, or the reason actually for that is I think that everybody thought we were innovative when we started the company. We were doing really high-tech biotech stuff, and we're only getting better at it. So every year since 2013, we've been named one of the 10 most innovative companies in the world across all industries. last year, I was very upset, and I'm not going to go into it. We were behind Tesla. There is really, I do not think that Tesla is innovative at all. It makes me question these whole rankings. I know we should be in there, but I don't know who does the rest of the rankings. But don't get me started. But it's all based on these technologies, and I'll briefly tell you about our major technologies from the beginning. And once again, I don't want to give you the wrong impression. I really think there's a lot of ways to make easy money in this business. Okay, but to build something that it really has lasting value, that really contributes and so forth, I think it really takes time. We didn't get our first drug approval for 20 years. We didn't become profitable till 5 years ago. Up until then, by the way, you should know this, the financial analysts literally used to ridicule us as the definite example of why you don't put scientists in charge. They said, like, look at this company, what a joke. It's been losing money for 25 years in a row. This is what happens when you put scientists in charge. And then miraculously, as Len says, we became an overnight success after 20 years of perceived failure. But now everybody thinks that this is the way to do it. And the way we did it was by building game-changing genetics technologies. Oh, I got to point this out. I'm sorry. These two slides or these two pictures of me and Len, very carefully chosen. Who's good at pattern recognition here? Got any AI specialists here? Okay, there's a pattern here. It's very obvious. Okay, it's very clear who's been carrying who on their back for the last 30 years. Now, I'm kidding. Len is a great guy. We would not have done a single thing, obviously, without Len. But I do, give him a hard time all the time. Okay, so these are our 7 approved drugs. I'll just touch upon maybe one of these stories to highlight them. But the way we do it, it's a very simple formula. I would like to think that it's the way almost any smart group of people would choose to operate. Okay? And we started this from the beginning. In fact, I was just talking with Rebecca Brown, working with some of her colleagues in the orphan disease space, and a long time ago now, looking for humans with mutants that either had superpowers or, were susceptible to disease. So protective and susceptibility genes, the old X-Man approach. So we started doing that the old-fashioned way back in the days of, maybe Rebecca's too young to remember this, days of like cosmid cloning and all this, when it'd take like 10 years to like map, the human gene cause of a particular disease. But what we started doing was revolutionizing initially the mouse genetic and engineering approaches, the notion of being able to genetically change rodent models, mice and rats, including genetically humanizing them to put in any mutation that you want. And in fact, to humanize vast portions of the mouse genome, as I'm going to show you. That really, together with the genetic targets, really empowered everything we were doing, because we would get the mutations, we would essentially knock them in before anybody else could knock them in into a mouse. We would reproduce the disease model in the mouse. That would start us on a path. We would put a therapeutic into now this humanized model, genetically humanized model of the disease. If it worked there, it should work in the humans, and that's why our success rates are so high. It's really so obvious and so trivial. It's amazing to me that other people haven't sort of kept up. And in the last 5 years or so, initially starting with, you all know Michael Leonardo, initially starting with some conversations and so forth with him, actually, that he helped inspire, we've been revolutionizing our human genetics approach so that we can feed the hungry beasts that we built more efficiently by getting many more human genetic targets than we used to the old-fashioned way. So these are some of the game-changing technologies that we built. So it's like Steve said, we build the technologies that allow us to genetically identify and validate targets and then test our therapeutics. And we also built genetic models that produce fully human therapeutics themselves. So I'm going to just very briefly highlight or give you the example of those sorts of technology. The first one we call Velocigene. We started this literally in day one when we opened the labs 30 years ago. And it's a way to do genomic engineering of the mouse. And we started by doing just simple knockouts and knock-ins. And now it's evolved to something that even today, with the use of CRISPR and so forth, I don't think anybody else can do. we can replace precisely to the nucleotide megabase. Megabase, that's about 1% of the human genome at a time, portions of the mouse genome precisely with their human counterparts to create unique humanized models, which I'll briefly get at. So like I said, it started at the beginning. One of the first people that I recruited to Regeneron was a guy named Tom DiChiara. He made the first knockout in academia as a graduate student, so I brought him to Regeneron. He made the first knockout in biotech history. And I still remember back then when Len and I were going around raising money, I had this quote on my slides when we were trying to raise money, that the company that best controls the genome of the most important laboratory animal model, the mouse, will become a leader in the biotech industry. Think I got any money for that? You know, people looked at me like I had 3 heads, and they would say, we don't understand. Where's the product? And I said, we're going to use this to, like, figure out which products to make. And they said, we don't understand. You're going to figure out which products? No, you need a product. You need to find one product. That's how most companies are built. One product, one idea. And by the way, try not to, please, go to those sorts of companies. Those are the vast majority of companies. It's one idea. It may be a very good idea from one academic. The problem is, I just told you the odds. If there's 5,000 companies and there's only 10 first-in-class medicines every year, how many of those 5,000 ideas that get started ever actually lead to a novel, new medicine? Very few, about 1 in 500 or 1 in 1,000. So you don't want to be going to a one-idea company. But that's what they want. Why? It's all statistics, okay? Most of the successful companies were one idea, one product companies. And people look at that and they say, oh, that's how you're supposed to do it. Look at this company. They had one idea. They devoted all of their resources to it. And finally, after 15 years, that one idea turned into an important story, an important drug. So if that's where most of the successes come from, is that the way you want to do it? What's wrong with that? You're suffering from the multiplicity argument, like when you do statistics or anything like that. They're ignoring the fact that at the same time that one company did exactly that, there were 4,999 companies that did exactly the same thing. And because they all failed, nobody's taking those 4,999 failures into account. Nobody's saying, well, this one worked, yeah, but these guys took exactly the same approach. They all failed. So your odds are 1 in 5,000. So basically what it means is that just because that approach works, it works really badly. That's why the whole system is broken. And what we got to build is systems that actually work much more reproducibly, which is exactly what I'm telling you about. But nobody got it. They didn't want to hear about how to build a system that can crank out opportunities, knowing that most of those opportunities would fail. They wanted one opportunity to know whether they could bet on it. And so the way we started out doing this, by the way, these are old pictures, but they're really cool, so I figured I would show it to you. This is how we built our Velocigene technology, as we say. Okay, this is actually a picture of me looking at a glowfish about 55 years ago. No, it's a reproduction. It's intended to be me looking at a glove. No, I just took this off the internet. Okay, that is actually a picture of me holding a glowfish. And so what we did, like 25 years ago now, is we figured out a way at the time to put in genes from one species precisely into a special location that allowed it to be expressed everywhere in the mouse, into the mouse. So this was the way we helped develop this into a high-throughput technology, because it was such an easy assay, how to rapidly, in a high-throughput fashion, precisely place a species from one gene replacing the corresponding gene or related gene in a mouse. By the way, just parenthetically interesting little tidbit. So we sort of became a little famous about 25, 30 years ago when we were doing this thing. And some guy calls me up and say, we're making a movie. And we hear that you guys are leading the way in trying to figure out how to move the genes from one species into another species. And we're making this movie. Anybody have any idea what the movie was?
Jurassic Park?
Very, very good. So we are a smart audience. Nobody ever answered that question before. Sign that guy up. Or did you hear me talk before? So So basically, they said, we're trying to figure out how to dinosaurize like lizards or something, and we want to make it as realistic as possible. Can we come visit? I had no idea who this Spielberg fellow was. I said, sure. So they came and all that. So those storyboards and all that, that is all, if you looked at them, if they ever paid attention, the original Jurassic Park, it's pretty real. And it's exactly what we do. But anyway, so you could, the problem is, it was their idea about where to get the DNA to start with. About using this fly. I think it was the author of the first book. I think it was Michael Crichton who came up with that. But that is a problem still. But that's a little detail. But anyway, so, this somehow contributed to Jurassic Park 2, as well as ultimately to billion-dollar drugs. I don't know which is more important. Okay, the Jurassic Park seems to get more attention. Anyway, one spectacular thing that we did with the ability to put genes from one species into the other is we had an idea a long, long time ago, okay, before any of you were born, okay, that what if you could make a mouse with a genetically humanized immune system? And the technology to really do that well did not exist when we had that idea. And we used the VelociGene technology to make a mouse with a genetically humanized immune system, which 6 megabases of mouse genes were precisely replaced by their human counterpart. That is I'll show you a little bit more about it. That's the most valuable biotechnology ever created in terms of both the dollars and the drugs it's produced. So basically, I like it, A, because it was my idea, B, because I couldn't have written it any better. But when I was with my mentor, Fred Alt, we wrote a paper based on some work we were doing at the time. And we said, one could imagine, for example, engineering a mouse to make specific human antibodies. Such a mouse might even be used to optimize such an antibody. In the most easily conceived version, something's weird about in the most easily conceived version, germline heavy and light chain variable region genes could be introduced. Although conceptually outlandish, such genetic program experiments may actually be realized in the not too distant future. Took about 20 years, which is relatively a short period of time in this business. But we actually did it. We meaning Drew Murphy and Lynn McDonald and Sean Stevens, Margaret Caro, in my group. And as I said, most valuable biotechnology ever created in licensing and collaboration revenues over $5 billion. We used to sell a pair of these mice, think about it, for $120 million. Okay? But more importantly than that, all the therapeutics that are coming from it. Already billion-dollar blockbusters coming from this technology. It was one of the things that Forbes featured. And it's something that obviously is a critical part of everything that we do. So this is what we do. It's really this simple. We start out with X-Men mutants. This guy feels no pain. This kid, 6 years old, incredible, musculature, right? So we literally have worked, with, in many cases, almost all of these cases, with outside collaborators to help map the genes. Once we think we have the gene, that's the key thing. When you're mapping genes, you're not sure you have the gene. How do you know you have the gene? So you knock it into a mouse and you prove. So in terms of this kid, okay, we put that gene into this mouse. Now, this mouse looks bigger. Obviously, it's a little hard to tell. If you're squeamish, don't look. But if you take the fur off the mice, it's pretty obvious. This is the normal mouse on the left. This is the mouse on the right. It's really sort of jacked. And we actually, so this is literally all we do. We work with outside, academics and our internal capabilities to map the genes. We prove we have the genes. We create essentially the animal model that we want to prove that we have what we want. In some cases, in this case, the pathway itself is what we want. In some cases, we want to block what we're putting in, as I'll show you in the next slide, I think. And we've already made antibodies for actually both of these targets that have actually worked in advanced trials in humans. Okay, so basically, we have something here for pain that really has, right now, it beats, here to the opiate crisis, beats opiates, NSAIDs, all that, in head-to-head studies for pain. And in this case, we've gotten, after a single injection, 8 to 10% muscle hypertrophy in human beings on average. who wouldn't want to take that drug? I don't know. I'll have to see the safety profile, but okay. So this is what we do. So those are cases where, you know, the last case was we have an antibody that mimics the mutation. Here you'd want to block the mutation. And I just show this because it's another major collaboration with Fred Kaplan at the University of Pennsylvania that led over many years to find this mutation. It's for a very rare orphan disease. It's a horrible disease known as fibrodysplasia ossificans progressiva. These poor kids, wherever they might have a bruise and a little injury, the inflammation activates the wrong process and they get bone. So this bump here is bone. This bump here is bone. This bump here is bone. And unfortunately, all these kids die because bone covers their entire skeleton. And think about it, it covers their rib cage. And their diaphragm ossifies too. So basically they can't take a breath. And you can't put them on a respirator because there's no room to expand. So, you know, it's a horrible death by asphyxiation. So once again, like I said, working with Fred Kaplan, we identified the gene and the pathway and all that, and then we humanized the whole thing, which is a very complicated humanization. You can see here's a normal skeleton. As soon as you turn on the bad mutation, you see this bone forming where it shouldn't, right at the knee joint, and in high power view, it literally, this is exactly what happens to the human. The bone essentially fixes the skeleton in place so you can't move. This is the lower leg bone, the upper leg bone, and the pelvis. This is all locked together. So tragically, these kids are locked in place with their skeleton. Eventually, it happens to their rib cage and their diaphragm, and they asphyxiate. The good news is, actually, because we were able to create this model, we were able to identify exactly the pathway involved. It's really a whole crazy story. You can read the published paper a couple of years ago. But once again, the good news is a VelocImmune derived. So we use VelociGene to create the model. We use VelocImmune to generate a mouse that either mimics the mutation or blocks the mutation. And that's what we got here, and it's already in clinical testing. And actually, within the next 6 months, we should know the results in this horrific rare disease. Okay, so this is the story of Regeneron, okay? We make all these technologies, VelociGene, ability to genomically engineer the mouse and specifically to humanize it, to create human models and test mutations. VelocImmune to create therapeutic antibodies that either mimic or block the mutations in most cases. That's led to our pipeline. Okay, as I said, very briefly, in the olden days, human genetics were these long collaborations. The Fred Kaplan story, I think, took like 15 years or something. All these stories took a long time. Now, with high-throughput sequencing, we've changed everything, and we've changed the timelines. As part of, Steve already mentioned Geisinger, also the US Biobank. We have these two major collaborations where essentially we're sequencing on the order of millions of people. And what's the value of that? Unlike other people who are being sequenced, all of our humans, so we've already sequenced 5,000 humans linked to their detailed medical records. That's the key thing, okay? And once again, I don't understand why this hasn't been done elsewhere, but this is really the key. I mean, this allows you to do human genetics at scale. Okay, people talk about big data. In the life science industry, there is no genetics big data that's worthwhile, because just sequencing people really has no value unless you know everything about that human being. And if you have a large amount of it, then you have big data that you can computationally study for the link between genetic variation to biologic variation. And oh, by the way, we have imaging. So we have total body imaging with the UK Biobank. And so we're sequencing all of these populations, and we're linking it here, and we're already making discoveries. I did want to mention, I think this is a great thing. By the way, the NIH, disappointed to tell you, has talked about doing this longer than we have. Francis Collins started this effort. It's been rebranded and renamed All of Us and relaunched. I think as of, they've been at it for at least 6 years now. As of at least, I think, the beginning of this year, you know how many people they had actually sequenced? 0. Okay, and they had spent hundreds of millions of dollars a year. I hate to tell you guys, but the NIH should be supporting innovative, individualized science. The NIH is not very good. Don't get duped by this at doing big projects because they just don't know how to do them. This is one place where companies, really do a lot better. So as a society, we've all probably spent $1 billion sequencing nobody. We've already sequenced half a million people with DI, all linked to their health information. But the beauty of is that we want to make it publicly available, but our shareholders wouldn't be happy with that. So we decided to have this entire effort co-funded by a partnership between a lot of the major biopharma companies in the world. And unbelievably, took me and Eris Barros, who's the head of the Regeneron Genetics Center, about 2 weeks, just call these people. They all said, sure, we're in. So because this is entirely being funded by biopharma, all the data will be openly available to the global research community. It already is the largest database of its kind. We hope it will have profound effect on human health. Oops. Okay. And it will be the first publicly available large-scale big data database linking human sequence to phenotype. So we're already using it, by the way, in the early stages. Right now, we're at the point of having sequenced internally 500,000 people. Most of them are coming from databases outside the UK Biobank. We have about 100,000 from the UK Biobank. We're already using it, and we'll be rolling it out publicly very soon, but we can already make big discoveries. I don't know if you guys know what PCSK9 is. It's considered one of the biggest discoveries in the field over the last 20 years. We had the first PCSK9-based drug. I'll tell you about it very briefly. We're already coming up with the next PCSK9, this one for liver disease, where we identified exactly what we want. Men and women who are protected from chronic liver disease. This is alcoholic, fatty liver disease, viral liver disease, and also toxic liver disease, bless you, from toxins. All of them are protected with this mutation. So it shows that this whole thing could work. And we actually have been working with a company called Alnylam. And very soon we can actually be able to put in a drug for this target. This is not an antibody target into the clinic. So it's definitely speeds up things. So it's all about these technologies, VelociGene, mouse genetics, humanized models, VelocImmune, the ability to make these fully human antibodies in a mouse with a human immune system, Regeneron Genetics Center, and they just really work together to really allow us to have the pipeline that we have. More efficiently than any other companies, we can put new drugs into the clinic that have actually been pressure tested in genetically humanized animal models. It really explains very simply why we have such great track records and success records. So this is what we've been doing for 30 years, and we're just trying to do it better and better, and we're not giving up on genetics. People ask me all the time, financial guys all the time, well, what's the next big thing? It's genetics. It's just about figuring out how to do genetics better and then apply your learnings. So there is nothing, I don't think, in terms of life sciences, I don't think there's anything bigger than genetics. Okay, and these are just 3 examples of going from genes and mutations. This is the one that I mentioned with some of Rebecca's colleagues at the NIH. But this is this story about PCSK9, which won a breakthrough award for our collaborators who found who mapped the human gene. But we developed the first therapeutic based on this work. I'll very briefly tell you about this, and then I'll end with largely focusing on a relatively new genetic story for allergic disease. So all I want to say about this is the PCSK9 antibody. Started with this great genetics story from our collaborators, Helen Hobbs and Jonathan Cohen, that won the Breakthrough Prize. And basically, we just announced a few months ago that we had finally tested this approach in what you have to do. You have to do these huge outcome studies with 20,000 patients studied for over 3 years, or 3 years or so. That's how you have to prove on, are you really reducing the most important thing, which is the risk of death? And this is the first study since the statins by Roy Vagelos that have actually shown a improvement or a reduction in death. And actually, they reduce death by about 30%. That is, they're saving about 1 in 3 people. But this is on top of maximally tolerated statins. So it really is an improvement. So this story just came out in the New England Journal of Medicine, and we're hoping that the FDA agrees and puts this in the label. and this will become, I think, the new standard for people who are at high risk from heart disease, that if you're really at high risk of dying, you not only get your statin, but now you get this layered on top. So that was a great story, and it proved the hypothesis that adding PCSK9 inhibitor on top of statins can result in marked reduction in coronary artery disease and death after less than an average of 3 years of treatment. The reason I have that in there is the longer you treat, the benefits should only get bigger and bigger. So I wanted to end with our last little gene to disease treatment story, which has just been coming out over the last year or two, which I think is really pretty spectacular. I'll let you be the judge, but it started in the same way. I'm not going to get into all the details, but genetics in humans and mice, that this could be the pathway that's the basis of essentially all allergic disease, and the results have been stunning, as I'm going to briefly show you. But basically, you guys probably all know that allergic diseases have been really ramping up over the last few decades. When I was growing up, there was no such thing as peanut allergies, right? We never heard about peanut allergies. Now, it's like you can't go on a plane with peanuts, you'll be like arrested. It's like terrorism, okay? These things are going crazy, and there's many different kinds of allergic diseases. The two biggest, well, not the two biggest, the ones that are thought to have, the most significant morbidity impact are in asthma, allergic asthma, and this skin disease called atopic dermatitis, where you get this horrible itchy rash over your body that's very much like poison ivy that doesn't go away. But there's food allergies, there's nasal polyps, there's allergic rhinitis, there's, like hay fever, there's now more and more allergic diseases being defined all the time, including ones that we never even knew existed before. So what do you do about this? So like I said, starting on genetics, my longtime colleague Neil Stoll and I had this very simple idea that was entirely based on genetics, that despite the different organ tissue manifestations, the one is in your lungs, one is in your skin, this is actually in your GI tract, and so forth, this is in your nose, okay? They're all exactly the same disease. They're all linked by the same mechanism, excess production of two interleukins, interleukin 4 and 13, and I started this collaboration with the very famous Bill Paul, who was a major player, who was a great guy at the NIH, and I met him in the 80s. That's when, in 1982, is when I started working on this project. And when Bill Paul at the time had identified interleukin 4, it was called B-cell stimulatory factor 1, BCSF1, and he hadn't even cloned it yet. And the reason I got involved with him, or I was interested in it, is because he could add it to B cells, and B cells would switch to making immunoglobulin E. Immunoglobulin E, for all those of you who may not know it, is the thing on mast cells or whatever. It is fundamentally, I think, the cause of most of allergy and allergic symptoms. And I remember joking with Bill Paul and my mentor Fred Alt at the time. We were going to try to study to understand how it did that. And nobody dreamed about blocking these things back then. But we literally joked with my mentor and Bill Paul, wow, if you could block this, maybe you could cure allergy. And as you may know, Bill Paul tragically died about a year ago now, or 3 years ago now. But he wrote, unbelievably, his last paper was a review ending with this story that I'm about to tell you, which had still not been approved, and he had said this is like a great story, and obviously he had a major role in it and all. He goes, the only disappointing thing related to what I told you guys, how long and hard the road was that it started literally 30 years ago, and it's still not approved now. But it's a great story, and Bill Paul was a great component of this, and our earliest collaborator on this. But in any case, we have now tried this approach, this VelociMune. So we use VelociGene to validate all this, genetic models, prove that it all works. We use VelociMune to make an antibody that blocks these two interleukins themselves. We've tested in 4 allergic diseases right now. Unbelievably, the results are crazy. This is not how drugs work, by the way. I don't know, when you hear about the biggest biologic in the world right now, for inflammatory diseases, Humira, okay, for rheumatoid arthritis. It causes an average improvement of 20% in 60% of the patients. And it doesn't do anything in the reigning behavior. So 20% improvement, 60%. Okay, this drug in this very severe atopic dermatitis condition, in 3 overwhelming, an average of 70 to 80% improvement in all the patients. And this is in the most moderate to severe patients, and it affects both the visualization of the lesions and also the itch. This was published in a series of papers in the New England Journal of Medicine. Same thing in asthma. Once again, drugs don't work like this. It's all based on genetics-based drugs do work like this. Okay, 66 to 81% reduction in asthma exacerbations with substantial improvement in lung function. Once again, these were papers that were just published back-to-back earlier this year. And these things first led to the FDA declaring that these drugs were breakthrough, which meant a major improvement for a major unmet need. Then the first approval was in atopic dermatitis. The second approval just last month was in asthma. And we also have two positive phase 3 studies in chronic rhinosinusitis with nasal polyps and eosinophilic esophagitis. And so the list, I hope, is going to go on and on. But, well, what I was going to do is say, so all these diseases are really responding. The response rates that I'm talking about, these kind of improvements, these are not what you traditionally see with drugs. But I think it is what you see with genetics-based drugs, not only ours, but there's very few of them. But I think this is why there's reason for hope for the future. But like they say, a picture is worth 1,000 words. Okay, this just shows a poor, and atopic dermatitis, severe atopic, is relatively, it's not common, thank God, but there's literally millions of children who are affected by this. So, and as I said, this is this horrible rash that covers over half for the moderate severe patients for which we're approved, covers more than half their patient, and it's a horribly itchy rash. I mean, they literally have to tie these kids' hands down so they don't excoriate themselves like when they're asleep and stuff. So I got this email from this girl's physician who said, with the family's permission, I'm sending you this. The family was in crisis. This girl was essentially hospitalized. She was miserable and would not even interact with the hospital staff. Okay, and this just shows, it's horrible, right? After a couple doses of dupilumab, wow. Okay, and like I said, it was on the note before, but the point was is when you do something like this and when you're miserable like this, you can't function, you're not a normal human being. This girl got her whole life back. And so, as the doctor said, after 3 doses, she's a pistol. So, as I said at the beginning, why do we all go into science? So we can use the power of science and we can do stuff like this. And, we have some crazy approaches in cancer where you have similar incredible responses and all this. So, but that's the big picture. So hopefully we have some time for questions and so forth, but that's the whole point. I believe that if you just stick to the basics, if you read Roy Vagelos' book, if you understand the fundamental principles, in this day and age, use the power of genetics, you improve the tools, you increase your chances of both better identifying the right targets and then coming up with therapeutics that better address those targets. All of our drugs to date, and somebody asked me this during the cocktail hour, all of our drugs to date, both approved and almost all of them in the clinic, are biologics-based drugs, but we are firm believers in the next generation of genetics technologies. We realize that all those approaches are not best done in-house. We have a major collaboration with one of the top 3 gene editing companies, Intellia, to incorporate gene editing in these approaches. We have a major collaboration with gene delivery companies. We have a major collaboration with Alnylam, which is an siRNA company. We have a major collaboration with one of the top CAR T companies, which is Bluebird. So I think it's a matter of different folks developing, and these are all gene-based or genetics-based approaches and technologies to therapeutics. We add in our genetics-based approach to target identification and validation, and now we have a choice of, we use Velocimune for antibodies, we use Alnylam for siRNA, we use Bluebird for CAR T, we use Intellia for gene editing, and we believe this is the present, and we hope it's only going to get better, and we hope that more of these efforts that are based on the genetics are going to accelerate the pace of drug discovery, because we need it. But as a society, we also have to recognize that we have to invest. Okay, these companies that I just mentioned were all newer startup companies that were able to raise money. Why were they able to raise money? Because somebody thought, oh, that company could become the next Genentech or the next Regeneron. So we want to invest in it. Okay, we have to keep that model going because right now there is no other model. And you know, I'm a firm believer that it starts with the fundamental basic research done at small scale in academia and at the NIH. And the NIH should not be trying to do what, you know, what biotech and biopharma do, like these big projects, because they've been proven not to be able to do this. This whole notion of translational research, it's a scam. You know, it's designed for like congressmen to buy into, okay, because they can understand it, but that's not how science works. You guys should all fundamentally be working more, you know, more likely to be working on fundamental processes in a worm or in a fly, or really fundamental processes having to do with human biology as opposed to trying to do translational work. And then translational work should be funded by society in a different way. And that's what we try to do. Anyway, that's it. That's enough of my philosophy.
Thank you very much. We wanted to bring up Hannah Mason for a few minutes to moderate a couple of questions. We're probably only going to be able to do maybe 3 or so questions, but you'll be around for dinner,
Sure.
and I'm sure you're available. So here you go, Hannah.
Okay, well, thank you, Dr. Yancopoulos, for that inspiring and entertaining talk. And Jonathan, amazing on the Jurassic Park reference. Okay, so now we'd like to open up the floor to questions. Nick, I saw your hand first. Wait for the mic.
Yes, very inspirational, an amazing story. And so, but I was really curious when you said 25 years of losses. So what do you say to investors in year 20, like, no, we're really close, like, trust us? Like, what do you say?
Well, so that's a great question. And I guess I should explain a little bit about, you know, how we funded, you know, this effort. So part of it was from the public markets, but mostly what it was, we eschewed, whatever that word means, the venture capitalist route, okay, because when you get venture capitalists involved, they tend to want a near-term exit strategy, as they call it, and they're just interested in return after a few years, okay, and we knew that we didn't want those, you know, time frames. And when I say we, I have to give huge credit to my longtime partner, Len. I mean, he was the one, you know, we like to think we're just as visionary in the business as we are on the science, and I like to think I'm a little bit more visionary in the science, he's a little bit more visionary in the business. So we eschewed, you know, venture capital money. We raised some money in the public markets, but the way we really did it, which I think was really brilliant, was when we had major scientific discoveries, the way most companies do it is at a very early stage, when they really think that they're onto something, they now go to Pfizer or Amgen or Merck or whatever now, and they sell out. And basically the company's sort of gone. They, you know, they get a lot, what seems like a lot of money up front. They get $100 million. The couple of guys who started the company, you know, make a decent amount of money, okay, but the company's sort of done, okay? And it's all up to the big company what happens to it, and most of those things die. Okay, our notion, and like I said, this was really innovated and invented by Len, was he said, you know, Amgen or Pfizer or, you know, Novartis or Sanofi, which is now Bayer, which are all partners in this, is you want this? Okay, you better want it really bad, because you know what you're going to do? If you want a piece of this, you're not going to get all of this. You only get 50% of it. So we're going to keep 50% on the back. So if this turns into a billion-dollar drug, we've become a big company all of a sudden, because we get half of a billion-dollar drug. That's what you need to make it. And not only that, how are we going to fund our work for the next 5 years or 10 years that it'll take to do this? You're going to pay 100% of that. So you're going to pay 100% of our R&D costs, and we're going to get half of the profits at the end of the day, because if this is a cure for eye disease or for allergies or cancer, it's going to be worth it, and that's the only way you get it. And so he innovated that model. That's what we did. We kept coming up with these discoveries. The remarkable thing, unbelievable, is some of our partners, like Sanofi, stuck with it and are now reaping the benefits of billion-dollar drugs. Some of them, just to show you the time frames and how these people think, 5, 6, 7 years into the process, they said, wow, this hasn't delivered yet. So they essentially gave it back to us. We only had maybe 2-3 years left to figure out how to fund on our own, and these guys lost out. So Eylea, which sells, think about this, around $8 billion in sales worldwide. There's very few drugs that actually do this. $8 billion, and in the United States where it sells $5 billion, 100% of it comes to Regeneron. Why? Because first it was owned by, not owned, but it was shared 50-50 with Procter & Gamble. They gave up on it after 4 years. Then it went to Aventis. They gave it back after 3 or 4 years. And then it was so mature at that point that Bayer was willing to fund the remaining couple of years of research, but it was looking so good at the time that they got only 50% of the ex-US sales, and we kept 100% of the US sales. So that's how we funded the business, based on our getting partners to fund our R&D and still give us 50% share at the end of the game. So this is how we now have billion-dollar drugs that we either get 100% of the profits because they gave up, or 50% of the profits, which is still pretty amazing.
Okay. We'll go to Joanna at the back.
Hi, thanks for a great talk. As one
Great
of the
accent.
Thank you. As one of the people who is the cause of the no nuts on a plane, how close are we to a cure for peanut or other food allergies?
Well, that's a great question. So we are actually, I think we are as close as our being able to actually show it. I mean, I think this molecule actually does it, though we have to prove it definitively. We actually, so obviously if you have people who have allergic atopic dermatitis, some of those will have food allergies. If you have allergic asthma, some of those will have, and we have, not for everybody, but an astonishing amount of sort of case reports and studies. I think some of them are even published, not by us, but by the individual investigators. What we know is, I'm sure you're familiar with desensitization therapy. So when you do desensitization therapy, it's no different in a man or a mouse. The whole key thing is the people who have allergies, including me, and it sounds like you, okay, for some reason in response to some challenges, often inhaled or food allergies that are absorbed generally through surfaces. It tends not to be things that you inject into you, okay, though that happens as well sometimes. But you make an IgE, immunoglobulin E response. Okay, as I told you, Bill Paul discovered 30 years ago that IL-4 is the thing that makes you make IgE. When you get desensitization therapy, have you tried or had desensitization? Okay, what they do is either by mouth for a food allergy or by direct injection in the skin. They give you that allergen, but they give it in an adjuvant. You guys all know what adjuvants are? An adjuvant that tries to induce an IgG response. And the whole thing is normally what you should generally have only in your body, unless you're infected by some parasite, is IgG. And normally you should have very low IgE. What happens is people who have allergies, specifically to that allergen, have very high IgE and very low IgG. So what the theory was 30 years ago by Bill Paul, which was abandoned by most people afterwards because nobody ever tested it, was, oh, it's not really this simple, it's like T cells, or it's something else. I don't know, they had all sorts of theories. Well, we know, and I'll tell you how, that it's totally a matter of just dropping the IgE and raising the IgG. And desensitization therapy, people haven't known exactly how it works. But one thing that it does is, over time, it takes a long time, because these people are allergic. When you give them their allergen, which you're trying to desensitize with, the IgE very slowly goes down, only in some people, and the IgG starts going up. Well, we did 2 experiments. We picked one allergen, cat allergen, only because it was relatively easy, and we had people who had this much cat IgE and this much cat IgG. And what we did is we just made, in our Velocimune mouse, we made IgG against cat allergen. Okay? And now we took these people and we just injected them with cat allergen. And we did this. These people, allergies immediately went away. So that proves, this was published in Nature something or other. So this proves that it is as simple as the IgG, IgE. So we have two choices. We could either make IgG for your specific allergen. That would be very custom and a lot of work for each specific allergy. We are doing that, but don't hold your breath. That's going to take a long time to get to different allergens. And peanut, for example, is multifactorial allergen. Okay, or what you could do is you could say, why don't I just block IgE production and keep cells from switching? So every time I'm injecting the allergen for desensitization, I make B cells that make IgG, and they stop making the IgE. So we've tested this in animals, and it works 100%. So it's very easy to make peanut allergic mice. If you inject them the old-fashioned way, it's very hard to flip them. If you inject them with dupilumab on board, these are fully humanized mice, they immediately stop making their IgE, and as soon as you inject them, you just ratchet up the IgG. And after a very short number of treatments, normally allergy desensitization only works about 50% after 2 years. In the mice, after like 2 injections, we're getting almost 100%. We are now in clinical trials for peanut and also for grass allergy. So all we have to do is everything that I told you is all still, in animals, extrapolated from other data and so forth, based on case studies, from dupilumab treated people. We just have to prove it, and the final arbiter is always the FDA. So just like we've got to prove for these other things, we have to do these studies. We have to get them done. We hopefully will get good data. We'll have to show the data to the FDA, and the FDA, we need, by the way, I'm a very strong supporter of the FDA. You want. You don't have to decide whether a drug works. You need somebody like the FDA. And people say, oh, FDA is slowing things down. No, FDA is keeping, snake oil salesmen from selling their stuff. So if we satisfy the FDA, then it'll be available. I anticipate that whole process to prove it will probably be within about 2 years. To get it approved will probably be another 1 to 2 years after that.
Okay, we have time for one more question. Jonathan, we'll go to you.
I think we got to get Sam in too because he was waiting. So, all right.
Right, so really cool, interesting, innovative pipeline. I'm curious as to sort of what the structure of Regeneron research-wise looks like from the inside. Do you run with a traditional, sort of head of lab or division with staff scientists and postdocs and people under them, or have you found some other model for structuring your research that works better for this pipeline for you guys?
That's a spectacular question. That's really important. I think that we have a totally unusual way that we structure our research. And in part, it was inspired by my experiences in academia. So I'll tell you this brief story. So my mentor was Fred Alt, who I already mentioned, who I think is a rocket scientist, one of the best in the world. And anyway, and he taught me everything I knew. I was very lucky. I was his first student. Okay, I was actually in the lab before, you know how it always works. He's supposed to come to start his lab, but it took him like 6 months or a year longer. And I, of course, had already committed. And I'm like in the lab trying to figure out what to do, call him up. And last thing he wants to do is be bothered by me. But anyway, when he finally came, I was all he had. Okay, and he taught me everything with his own hands. And I think that was so critical to my training. And we had a great experience. Then we had, at the same time, pretty much, there was another grad student and then another one. And it was a small group of 4 or 5, and it was a great environment and all that. And I stayed with him for 10 years because I loved it so much. And at 10 years, he had 30 postdocs working for him. And when I would come to lab, my stomach would churn. I went from my loving, all I wanted to do was be in lab, okay? Because my buddies, we were a family, we're all together, we're all in it together. When he had 30 postdocs in the lab, I knew that every other postdoc in that lab was rooting against me, okay? And that made me sick to my stomach, literally. And then I ended up getting, in the same department that I was in, I ended up getting a faculty position. And once again, I had this thing where they were waiting to finish the construction on my lab. and I was still working with Fred in his lab while I was waiting, still doing all these great experiments with Fred. And I got taken out to dinner with the departmental chair, and he goes, well, I just wanted to be clear about one thing, that once you actually start your own lab, you can't publish with Fred anymore. And I said, wait, the whole reason I want to be here is because I want to like publish it. And they said, well, no, there's this thing called tenure, and you have to prove that you're independent. Okay, so you have to be able to prove that you can do this without Fred. So I said, wait, this is exactly what I said. Wait, let me get this straight. If I can cure cancer with Fred Alt, that's not good enough. I got to cure cancer by myself. And he said, yep. And then I said, so it just happened that two of my heroes back then, who not coincidentally are now been on my board for 30 years, Brown and Goldstein. You guys heard of Brown and Goldstein, I hope? God, these are heroes. These are people who were trained at the NIH in the intramural program and then won Nobel Prizes. Anyway, okay, read up on Roy Vagelos, read up on Brown and Goldstein. Anyway, so I said, and they were famous guys who together won a Nobel Prize. So I said, well, how about Brown and Goldstein? Why can't me and Fred be like Brown and Goldstein? And I said, he looked at me and he goes, well, you're neither Brown or Goldstein. So it was sort of coincidentally and around that time, by accident, that I was approached by this guy, Len Schleifer. Okay, and also I have to say, the other big thing at the time, if you want to hear like a little personal story, my dad, who was a Greek immigrant, he was all for education and all this, and he was so proud that I went to the Bronx High School of Science, and I went to Columbia, and I was doing so well. And then he started getting really nervous and upset that I just kept going to school. And here I was, in my late 20s, and I was still working in the lab with Fred, making at the time, whatever it was, $8,000 a year back then, and he was saying, what was all this education for and all this? And he was saying, and I said, well, Dad, I think that I could maybe, use science to like cure disease. And he said to me, he was no dummy, even though he never finished high school, and he could barely speak English. And he said to me, well, I brought my family to the greatest, country on the face of this planet, which I still believe is true. And he goes, and you can do anything you want here. If you really think that you can actually cure disease, I guarantee you in this country, in the United States of America, you can make a hell of a lot more than $8,000 a year doing that. and literally, it was the combination of realizing that I was going to have to do it alone without Fred Alt, who, first of all, it took the fun out of it, and then it made him a competitor of mine, okay? And my dad sort of telling me that, there's another way. And then coincidentally, I literally got what was almost a cold call from this guy, Len Schleifer. Back then, I was one of the first, in Fred's lab, we were one of the first gene cloners. Believe it or not, the 80s was when they first cloned the first genes and stuff. So he was looking for a gene cloner, and I was like the only gene cloner who answered his phone calls. And it was a combination, and because biotech back then, it was like going to the dark side, and you were lost forever. And it was because of all these reasons that I said, I'm gonna do this. Now I'm ready to answer your question, because you needed to know that background. Okay, so when I went to Regeneron, I said I wanted to be like the early days with Fred Alt, when we were all one family, and we all worked together, we all support each other, and then I felt like I was going to work with like my friends, and that everyone, how's that saying go? All for one and one for all. I mean, we're all in it together. So we, Len and I, and also not only us, the first couple people that we hired, we said this is going to be our model. Okay, yes, people are going to have their own sort of groups, but we're not working in isolation. So all the heads of the groups, right now there's about, I would say about 30 to 40 groups. Okay, all the heads of the groups are going to get together on a daily basis for a couple of hours or as long as it takes to, on a daily basis, review each other's science. So, group leader one, you have 30 people that you have to sort of discuss your science with. And yeah, we're all critical and skeptical and all that, but the whole goal is, let's figure out how to help this person. And if I have an idea or I can do something in my group that can help that group, that's the whole point. Okay? So that's how we're structured. So we are structured somewhat traditionally like academic labs, but the difference, and it extends beyond just the group leader. So we have totally integrated meetings all the time. And the way we work is, in fact, some groups almost solely serve to develop technologies and to serve the other groups in a matrix way. So I believe that we have the world's best mouse geneticist, Brian Zambrowitz. I think there's a lot of evidence to say that that's true. Okay, he lives to improve and create better mouse genetics technologies, but he also runs the group that creates the animal models for all the other groups as a collaboration. Okay, and so they sit there and they say, well, how do we want to make this model and so forth? All of our groups work that way. So somewhat traditional. We have a very strong postdoc program, if any of you guys are interested. We also have like the Regeneron Prize for both graduate students and for postdocs. And if you get into the top 10 of that, we offer everybody a job, and everybody gets a monetary award, but the winners, or maybe the first two in each class category, actually get a $50,000 cash prize that does not just go to replace your grant. It goes into your pocket, by the way, okay, because we believe in incentives. So there's all sorts of things that we're doing to try to attract, young talent. So you come there, you are working in a group, and if you're, let's say, a postdoc or a young scientist, but in a very, very integrated way where everybody works together. And moreover than that is that in most companies, there's research, and then there's like clinical development and so forth. We have no walls. So if you invent a cat allergy drug, or if you invent dupilumab, or if you invent Praluent, and you want to lead, because we think that the scientists actually know more. Like MD PhDs do have an advantage because they have some of the clinical training. But if you're a scientist without an MD and you've invented the pathway or the drug or whatever, you have the right to cross over. So in other companies, they literally throw it over a wall, like a 10-foot high wall, and they never see their invention again. And they have no idea who the people are who develop it. You can actually be the lead, or you will definitely, we will force you to be on the team because we think that you know better than anybody else about how that should be used. So these are levels of integration and lack of walls that I don't think exist anywhere else from academia to any other company. So I do think that we're very special in terms of how we're organized, which makes it, I think, fun and enjoyable. We're all in it together. We're all trying to help each other, And of course, we're all a little competitive. These are all people who got, perfect scores on their SATs and so of course these are people who are highly driven and motivated, but the people who rise to become group heads they're selected to be people who have figured out the magic of not only doing their own science, but the reason you get promoted actually is because you can, think about this. Just think about this, okay? I'll make the analogy. Carmelo Anthony. Anybody know basketball? Yes. Worst guy to have on your team. He may be so talented, he makes the team worse. Okay, so you only get promoted, if you are a Carmelo Anthony, we literally, not only we don't promote you, we get rid of you at Regeneron. Okay, as soon as we can tell that you're that phenotype. We want the guys who, they can maybe score, but they're also a great passer, and they're a facilitator, and they do the dirty work, and they, box out onto the boards. Okay, if you can make your teammates better, you get promoted, you become a lab head. If you're a lab head that can't make the other lab heads better, you're never going to be a lab head. That's how we work.
Okay.
All right.
Well, thank you again, Dr. Yancopoulos.
Renaud from the Lasker Foundation, and I wanted to give you just a little something to say thank you for coming and for sharing your experience and all your enthusiasm for your company and for all your work. Thank you so much.
Great company to work for.